Thank you very much, everyone, for joining. It's my pleasure to host this EASD Academy Journal Club. It's the 4th edition, and it's partnership with Diabetologia. So it means it's going to be a new paper recently accepted for publication in Diabetologia to be presented, and it will be his first author, Mohammad Tariq, who have very kindly agreed to present his paper today. So just to briefly introduce Tariq, he did his PhD in Japan in quite a different field, more in cancer research. And then he moved to Europe to come to University Catholique de Louvain in Belgium as a postdoc, where he did his research. He's going to present us about the culture of islets and the response to high glucose during extended period of time for weeks. And then for the last couple of years, he moved to another postdoc position in Sweden, in Lund University, where he finished. He managed to get his paper accepted, and he's working on a new project, still working about islets and glucose. So thank you very much, Tariq, for joining us and agreeing to present your paper. And the floor is yours to present your research.
Thank you very much, Pierre, and good afternoon, everyone. First of all, I would like to thank the EASD Academy for providing me this opportunity to present my work here at this platform. It's really an honor for me, so thank you very much, the EASD Academy. About my paper, which was recently published in Diabetologia. The title of the paper is that prolonged culture of pancreatic islets under glucotoxic conditions changes their acute beta cell calcium and insulin secretion glucose response curves from sigmoid to bell-shaped. So this work was carried out at UC Leuven in Brussels, Belgium, although currently I'm in Lund University, but the work that I'm presenting today, it was done in UC Leuven in Brussels. So this is the information about the paper, the DOI and all the authors that you can see here. So as all of you know, that pancreatic beta cells maintain glucose homeostasis by metabolizing glucose, resulting in increase of ATP to ADP ratio, which leads to closure of K-ATP channels, resulting in plasma membrane depolarization and opening of voltage-dependent calcium channels, which trigger the secretion of insulin. Now, this happens in the normal conditions, but in diabetes, the beta cells become dysfunctional, and there are genetic and environmental risk factors which contributes to beta cell failure and insulin resistance, which leads to diabetes and chronic hyperglycemia. And chronic hyperglycemia in turn exerts further deleterious effects on the function of beta cells. So it has been shown in rodents that glucotoxicity leads to reversible changes in beta cell gene expression and glucose-stimulated insulin secretion. There are beneficial effects of beta cell rest on insulin secretion in rodents. In human beta cells in culture, they are less prone to apoptosis by glucotoxicity or by high glucose. So therefore, a decrease in beta cell function rather than number contributes to changes in glucose-stimulated insulin secretion in early diabetes. And therefore, the aim of this project was to understand the changes in beta cell function after long-term high glucose culture. So in order to study the changes in beta cell function after long-term culture in high glucose, we obtained human pancreatic islets and cultured them in different glucose concentrations, such as 5, 8, and 20 millimolar glucose for 1 week or 3 weeks. And these islets are named as G5 islets, G8 islets, and G20 islets. G5 islets in black, G8 in green, and G20 in red. And all the data is color-coded as well, so please pay attention to the colors. So we used to culture these islets for 1 week or 3 weeks, and then we used to perform some functional experiments. So today, the results that I'm going to present are only for 1 week cultured islets in these different glucose concentrations. So first, we looked at insulin secretion in the culture medium during the culture of these islets. So during the 1 week culture, we used to collect an aliquot of the culture medium after changing the medium, and we used to measure insulin in the culture. So as you can see that G8 and G20 islets as compared to G5 islets has significantly increased secretion of insulin in the culture medium. Next, we checked the insulin and DNA content on the left panel, you can see. So as you can see that insulin content of G8 and G20 islet is significantly downregulated as compared to G5 islets, while the DNA content remains the same. On the right panel, you can see that we measured the proportion of different islet cells. So here in this case, beta and alpha cells. So we measured beta and alpha cells in G5 and G20 islets, the black and the red traces. We couldn't see any differences in the proportion of beta and alpha cells after high glucose culture. So, and finally, we checked a beta cell apoptosis by tunnel and insulin staining of the beta cell after the 1 week culture in different glucose concentrations. And we found that after culturing in 8 or 20 millimolar glucose does not result in increased apoptosis of beta cells. So therefore, after a high glucose culture, the apoptosis of beta cells is not increased. So therefore, it might affect the function. So next, we checked the cellular metabolism by first measuring NADPH autofluorescence and ATP to ADP ratios of these islets after culturing in different glucose concentrations. So on the left panel, you can see NADPH autofluorescence. On the top of the panel is acute glucose stimulation. So we can see that there is a stepwise acute glucose stimulation as shown on the top of the panel. Black is G5 islets, green is G8 islets, and red is G20 islets. So as you can see that the NADPH autofluorescence, even in the basal conditions after 1 week culture in high glucose, so even in the basic condition, the NADPH autofluorescence is higher than G5 islets, but addition of acute glucose could further increase NADPH autofluorescence in these islets, although less than the G5 islets. On the right panel, you see the ATP to ADP ratios of G5 and G20 islets. So each, like G5 islets and G20 islets, there are 5 conditions in each. And as shown in the legend on the right, so the islets after 1 week culture were cultured 2 times in different glucose concentrations as shown in the legend. And then we measured ATP to ADP ratios of those islets. So as you see, the G5 islets, ATP to ADP ratios is increased by dose dependently by acute glucose. But the G20 ATP to ADP ratios, it remained high and it is increased by, the ATP to ADP ratio in G20 islets remained high. Next, we did some functional experiments to check the function of these islets. So after culturing for 1 week, we checked intracellular calcium and insulin secretion. So for intracellular calcium, we used a Fura2 probe. So we measured Fura2 fluorescence of these islets after 1 week culture. So The black is G5, green is G8, and red is G20 islets. So as you can see that the G5 islets, the black traces, they respond dose dependently to glucose, to acute glucose, as shown on the top of the figure. So the G5 islet responds to 10 millimolar acute glucose. While if you look at the green traces, the G8 islet responds to 5 millimolar glucose a bit earlier than the normal G5 islets, and then there is further response at 10 millimolar acute glucose, and then the response is blunted. If you look at the red traces, the G20 islets, the basal calcium is already high in G20 islets. Then there is a response to 5 millimolar acute glucose, and then the response is blunted. But interestingly, above 10 millimolar acute glucose, the calcium influx is actually inhibited. This inhibition of calcium influx is not transient, and therefore it was a human-specific phenotype, because this type of phenotype has never been observed in rodent islets before. And similarly, the dynamic insulin secretion on the right side followed a similar pattern as a calcium influx. So the calcium fluorescence that we saw through FURA2, we wanted to confirm that the calcium changes are occurring specifically in the beta cell. And to do that, we use a genetically encoded calcium sensor, D3CPV, under rat insulin promoter. And we could replicate the results that we found in FURA2. So the calcium changes that we were measuring, they are occurring specifically in beta cells. So as we saw in the calcium and insulin secretion, that the basal calcium and insulin secretion was higher. So, which prompted us that the beta cell plasma membrane seems to be depolarized, even in low glucose concentrations. And to check that, we measured the beta cell electrical activity. So to do that, we as you can see on the left panel, we cultured human islets on multi-electrode arrays, and we recorded a slow potential frequency. And slow potential frequency, these are beta cell specific signals, which are recorded by the electrodes. And as you can see that the G8 and G20 islets, the beta cell electrical activity is already high, as we saw in the calcium influx and insulin secretion. So electrical activity is already high, but in G8 islets, there is a further increase in electrical activity by acute glucose addition. But if you look at the red traces, when we go from 10 to 30 millimolar acute glucose stimulation, the beta cell electrical activity is actually inhibited, as we saw in the calcium influx as well. On the right side, in order to confirm that the beta cell plasma membrane remained depolarized, we used a diazoxide or a KTP channel opener to see that whether a diazoxide could reduce the higher calcium and insulin secretion in low glucose after culturing in high glucose. And as you can see, that's both calcium and insulin secretion of G20 islets. It went down after the addition of diazoxide, which shows that the beta cell plasma membrane remained depolarized after culturing in high glucose. Next, we checked whether the glucotoxic changes that we observed in beta cells are reversible. And to do that, we cultured these islets either in G5, either in 5 millimolar glucose for 3 weeks, G5, 3 weeks in G5, black traces, or the first 2 weeks in 5 millimolar and the final week in 20 millimolar, G5, G20, the red traces, or first week in G5, second week in G20, third week back to G5, the orange traces. So on the left panel, you see that insulin secretion during the culture. And as you can see that The black traces, the G5 islets throughout, the insulin secretion in the culture is low. If you look at the red traces, which are the islets cultured for 5 millimolar glucose in the first 2 weeks, 20 millimolar in the second week. So you can see that the insulin secretion in culture is increased in the third week. While the islets which were cultured in 5 millimolar glucose in the first and third week, but 20 millimolar in the second week, you can see that in the first week, the glucose, the insulin secretion is low. In the second week, when we increase the glucose concentration to 20 millimolar, the insulin secretion in culture medium is increased. And then when we reduce the glucose in the culture medium to 5 millimolar, insulin secretion in culture is reduced to back to the level of normal islets. Similarly, the calcium traces on the right side, you can see that after culturing for 1 week in 5 millimolar glucose, the calcium response is similar to the normal G5 cultured islets. So it means that the glucotoxic changes that we see, they are reversible. Next, we wanted to confirm that as we saw that the ATP to ADP ratios and NADPH autofluorescence increased upon glucose stimulation, and the ATP to ADP ratios in G20 cultured islets, they were higher, remained higher even in the low glucose concentration. So it suggested that after culture in high glucose, the glucose metabolism is increased, or the glucose metabolism is enhanced. And to check that, to find that the calcium phenotype that we found is due to increased glucose metabolism. So we cultured islet in G5 in 5 millimolar glucose, G5 islets, 20 millimolar glucose, G20 islets, or 5 millimolar glucose in the presence of glucokinase activator, GKA here. And on the left panel, you see insulin secretion in the culture medium. And on the right side, you see intracellular calcium influx. And you can see that just the addition of glucokinase activator replicate the results of G20 cultured islets. So we found that insulin secretion during the culture was similar in G20 islets as well as G5 islets cultured in the presence of glucokinase activator. Similarly, if you look at the calcium traces, so the addition of glucokinase activator to 5 millimolar glucose results in replicating the same response as by the G20 islets. So the intracellular calcium response is inhibited above 10 millimolar acute glucose, as we see in G20 cultured islets. So which means that the glucotoxic alterations that we see are occurring due to increased glucokinase activity and increased metabolism. So to conclude, the high glucose culture of beta cells result in changing the acute glucose response from the normal sigmoid to bell-shaped, and this response is reversible after normalizing the glucose concentration for 1 week. And I would like to thank all my lab members and the collaborators who helped in the studies, and great thanks to the pancreas donors. And I would like to take advantage of this opportunity to make one announcement. So we have one open postdoc position in Jean-Christophe Jonas's lab, the lab where I carried out this work. So if anybody is interested, can contact Jean-Christophe Jonas directly. Thank you very much.
Thank you very much, Tariq, for this very nice and clear presentations. It's a lot of data, but you managed to present it very well and make it very understandable, or I hope it will be. I will ask with a few questions, but then I will open the floor for the audience. So if you have any question, please think about it. And then you can either ask them in the chat or raise your hand, and then we will give you the microphone, or you will just have to open the microphone to ask a question. So please feel free. It's a very friendly discussion we should have now. And we are lucky to have Tariq to explain a bit more about his paper, about what he has done. So feel free to use this nice opportunity. My first question, Tariq, is about a little bit of background. How did you choose to come to do this postdoc in Louvain to work on this project, and how was it funded?
All right. Yeah. So first, I did my PhD, as you told in the introduction. I was doing my PhD in cancer biology. And at the end of my PhD, we were mostly focusing on cellular metabolism, mitochondrial metabolism, and we were like working on aging as well, aspects of aging and how mitochondrial metabolism is important in aging and those kind of things. So I sort of developed an interest in cellular metabolism, organismal metabolism. So therefore, I wanted to to move to metabolism field, and therefore I found this this position in Jean-Christophe Jonas's lab, which was really interesting. And I reached out, and I got selected there. So yeah, that's how.
Okay, and can you tell us about the funding? So you mentioned you had different agencies who funded, and what was your funding as a postdoc? So it was a position opened by Jean-Christophe Jonas?
Yes, this position was funded by Innoviris. It's the Brussels funding agencies, and also by the SFD. And it was a postdoc fellowship by Jean-Christophe Jonas.
It's good about opportunities and to know how you can have it. My next question is also still because you finished this paper, it was published 1 year after you moved to a different lab. Sometimes it can be quite difficult when you are a postdoc or a young researcher to finish your research from the previous lab. What is your experience about it?
Yeah, so yeah, I was in another lab, but we had a frequent contact. I've been in contact with Jean-Christophe since I moved, so we regularly contacted about the paper. And yeah, most of because there were no experiments needed to be done, I did all the experiments and everything, but yeah, the modifications and everything. So Jean-Christophe helped me a lot in that as well. But yeah, we have been in regular contact, and we have been discussing, and that's how we did it.
Okay, no, that's a good job. Congratulations for this paper. It's very interesting. And if I move a little bit more about the science, and then we can discuss about your career a bit more later. One of the striking effects you have is this kind of sustained insulin secretion after G20 for 1 week, even when you put back the cells in low glucose. What is your explanation, in your opinion? And is it an effect that will last a long time or not?
So the secretion is like the response that we saw, like the glucotoxic changes that we observed. So they are reversible after 1 week of normalizing the glucose concentration. So we believe that these changes, like these dysfunction after culturing in high glucose, is reversible after normalizing the glucose, although this is a very severe phenotype, but it is reversible, yes.
And how long does it take? Because you've only tested 1 week after. Do you think it's within few hours, or it takes days for the cells to change their metabolism and go back to normal?
I think definitely days. We checked 1 week, and if I remember correctly, we tried, not myself, but other people in the lab tried like a few days. So at least it takes a few days, I think more than 2 days. But yeah, not hours.
Okay. And have you tried, or as others tried, to characterize in terms of phenotype of transcriptome or proteome analysis of the state of the cells after this glucotoxic condition?
Yes, we have some follow-up of this study. So yes, we have some transcriptome and proteome data that but we still have to look at it, and we still have to validate some targets in that.
Okay, so a follow-up for maybe one next journal club, or at some point you will have a presentation. No, great. And looking if there's any hands. So if you have any question in the audience, feel free to ask. In the meantime, I have one also, because here you looked more specifically at insulin. In the pancreas, you also have the alpha cells producing glucagon, or even the D cells producing somatostatin. Do you think there is any effect of this high glucose on these hormones too?
So basically we were focusing, our lab is basically studying beta cell function. So we were mainly focusing on beta cells. So we didn't check glucagon, but we did check somatostatin, and there was no difference in somatostatin secretion in these islets. But since we were focusing on beta cells, so we mostly studied insulin and not glucagon and those other hormones.
Okay, oh, that's
May I ask a question? Because, hi.
Sure.
So I am a clinical researcher, so please excuse me if the question is silly. But so you exposed the islets to glucotoxic conditions up to 3 weeks, right? How does this translate into clinic? I mean, what would be the duration of diabetes, let's say, of uncontrolled diabetes in a human? How does that translate? Is it possible to, I don't know, imagine how that would translate into humans?
So, so first of all, the 20 millimolar glucose that we used. So first of all, that is a very high glucose concentration, so which is not observed in vivo, or maybe in most severe like undiagnosed and untreated diabetes, but it is very high, which never occurs in the diabetes. But in the diabetic people, it may take not days or weeks, but it may take months and years to develop, because and that's why the moderate condition that we have, the G8 cultured islets. So that we think is the tipping point from compensation to dysfunction, you know, because in G8 islets also they become more glucose sensitive as G20 islets, but they are still like the response to glucose in terms of calcium and insulin secretion, the amplitude of the response is increased, and so it means that these islets are compensating for the deleterious effect of high glucose. So in terms of like the physiological, so in physiologically, of course, it will take from months to years to have those worsened effects.
Thank you. And I also have another question, also somehow that somehow relates to clinical stuff.
Yeah.
About donors. I mean, because donors may be different, like different duration of diabetes, for example, or different BMI. Do you ever control for these characteristics in vitro studies? Because this is something I always wonder.
Yeah, exactly. Yeah, this is a very nice question. And yes, so we had donors. We obtained human islets from 5 different isolation centers. And we use like human islets from male and females, from normal and diabetes, with different diabetes condition, with different BMI and different ages, and etc. But the story that I was presenting today and the functional alteration of beta cells that occur, it is irrespective of the donor age, BMI, or sex. So it is irrespective of all those changes, all those parameters. So whatever the age or the BMI or the isolation center, or so all those parameters, they do not affect the changes that we observe.
How do you know?
We checked it in our paper. In my paper we have this data, but unfortunately due to time I didn't include those data.
Okay, thank you.
Yeah.
In your paper, you also, and you didn't present it today, some data about islets coming from diabetic donors. And so what's your feeling? Do you think the diabetic donors already had a kind of toxicity on the islet, a phenotype of toxicity, or was it, they were almost like the normal islets?
Yes, they had a phenotype of toxicity. They had cytosolic oxidative stress. And the response, like we cultured them for 1 week. So the response, the amplitude of the response was smaller than the normal islets. So although they responded after 1 week of culture, they responded to acute glucose, but the response was lower than the normal islets. And yes, they had functional signs of glucotoxicity, like they had oxidative stress, cytosolic oxidative stress.
I had a question. Mohammad, thank you so much for your presentation, really nice data. And I'm curious about the gene profile of beta cells. I don't know if you check the gene expression of beta cells under this glucose condition, and if you think that this adaptation that you are observed could be related with changes in the genes in the beta cell profile.
Actually, yes, we did gene expression, and we are working on it. But the changes that we observe are less likely to be due to changes in gene expression, because yeah, or maybe the activity of the genes, but yeah, because the changes are reversible. So yeah, maybe it might be the activity of different proteins or whatever. But that we have to look at it like for later.
And on the same kind of question about adaptation of a cell, did you look at the insulin production? So just maybe at insulin mRNA, but also how much is produced? Do you have unprocessed insulin, have a ratio of unprocessed insulin over processed insulin in your cells? Because you have this big drop in insulin content in G20 cells.
Yes.
However, they keep secreting more insulin, which is a bit, I don't know how to explain it. So I don't know if you have any ideas about what's happening in these cells.
So yeah, specifically the expression of insulin, we didn't check that in this study. But yeah, it definitely seems to yeah, to go down, because the content goes down, but the secretion is yeah, the secretion is increased. So like in the normal islet, the content is more, but the secretion is less. But here the secretion is enhanced, so it is down, but the secretion is high.
And do you know, have you calculated if the total amount of insulin produced, so if you make the sum between what is secreted and what is inside the islet, is the same between the conditions, or does it change? Is it something which is doable? I'm not working on islets myself, so it might be a very naive question.
No, we didn't check that. I'm not sure about that.
It's something it's because it's, yeah, I think which is very interesting to see how this change on the cells can adapt. And it's also this reversion, which I think is quite fascinating. You did the studies also for 3 weeks. And
yes,
do you still have reversion after 3 weeks? Or do you think there's some points where the cells will stop to be able to readapt and go back to a normal, healthy status?
So beyond 3 weeks, you mean if we culture beyond 3 weeks?
Yeah.
Beyond, no, we didn't culture more than 3 weeks. So I'm not sure whether more than 3 weeks will be reversible or not.
On a very naive question, because I'm not in the field, is it something which is doable? How long can you keep fresh islets alive? Have you tried to keep them, or in other experiments, or have colleagues trying them for longer?
So usually people don't keep islets in culture for that long, especially like for doing insulin secretions and those kind of experiments. So people like immediately after getting the islets in a few days, they use the islets because they say that if you culture them for too long, the insulin secretion response is not that good as it was in the beginning. So I think you can keep them for a couple of weeks because in our case, we cultured them for 3 weeks and yeah, we could see the functional response. But beyond that time, I'm not sure whether the response will be there, same or not.
And did you have to face technical challenges or to improve your protocols to maintain the cells for that long? Because it's quite an innovative thing, as you said.
Yes, yes, we used to adapt different techniques. So like in the beginning, we were using BSA instead of FBS in the culture medium, and we found that BSA, although there was no difference in human islets culture, but in rodent islet, the BSA leads to apoptosis, increased apoptosis in glucotoxic conditions. So therefore, we tried to use FBS for that, and then for human islets as well. So yeah, this is one example. But yeah, of course, we need to we used to adopt different conditions according to according to the islets.
All the small tips which can help also other researchers sometimes, which are very important. And I think in your paper, the methods are quite well described. So again, it's a very nice thing to be able to replicate the information.
Hello, I don't like to intervene without having the consent of Tariq, but I would like to have just to mention to Caterina Conte. I think, Tariq, you could have said to her first question that we tried after 3 weeks of culture in 10 millimolar glucose, we have the same effect as after 1 week at 20. So the longer the culture, the lower the glucose concentration at which you will see the phenotype that we describe. So that may be answering your question, that maybe very long-term moderate hyperglycemia may have the same effect in vivo, but this is going probably too far from our data, of course. And regarding the phenotyping, proteomics and transcriptomics, we've done RNA-seq, we've done proteomics. We have a lot of data also that Tariq contributed to, and that's why I'm looking for a postdoc to finish this. Thank you.
Thank you for this addition, and yeah, about the feedback, and yeah, seeing also this nice opportunity for a postdoc if anyone is interested in.
Speaking about opportunities, as people think of questions, I would like to remind everybody that we have deadlines coming for to apply for becoming a member of the EASD Academy Committee, and also for the EASD Academy Mentorship Program. So if people want to apply, they should do it by the 16th of April. So this is another opportunity, a lot of opportunities here today.
So feel free to check the Academy website for all the information about it. It's you will have, and you also have the recording. So today is recorded, and as well the previous journal clubs and some of our webinars. If there's no other question at the moment, maybe Tariq, can you tell us a little bit about what are you doing now? What is your study in your postdoc in Lund? And how did you get this postdoc?
Yeah, so currently I am working in Lund University Diabetes Center. And here our studies are focusing on the mitochondrial VDEC1, voltage-dependent anion channel 1, in diabetes, the role of this protein in diabetes. And this lab previously showed that VDEC1 is upregulated in diabetic patients and also in like glucotoxic conditions. This protein is upregulated, and this is mitochondrial protein, but in diabetic or glucotoxic condition, this is mistargeted to the plasma membrane of the beta cell and resulting in ATP loss and beta cell dysfunction. So I'm further working on to reveal the mechanism of this mistargeting of VDEC1 to the plasma membrane, and how is it involved in the dysfunction of beta cells. So yeah, broadly, this is what we are focusing on now here in Lund University.
Okay, and is it something are you using also the data or what you've learned from the previous postdoc, especially the glucotoxic effects on the long term? Do you think it's one of the mechanisms that could explain the phenotype you observed in this paper?
Yeah, yes, of course, that might be yes, that might be one of the the phenotypes that we can check here, but we are not checking that phenotype at the moment now, or like not that long-term culture that we did in the previous postdoc. But yeah, definitely the previous postdoc really helped me to like again the techniques and learn all the techniques that I'm currently using, because in the previous postdoc in Jean-Christophe's lab, when I joined there, I didn't know much about the islet and beta cells, and I didn't know all the techniques that I presented today. So I learned everything there, and yeah, Jean-Christophe really helped me a lot in like also technically and theoretically. So yeah, of course, I'm taking advantage of that now. So it really helped me. Yeah.
And it's nice to see how you can come into this field and learn and manage to do all the things you've done, because it's a lot of work, a lot of data you published in a very good journal at the end. So congratulations for managing to do all that.
Yeah,
it's not easy to learn all that.
Thank you so much. And yeah, I think I'm very thankful to it depends on the PI. When the PI is cooperative, so everything is possible. And I really owe a lot to Jean-Christophe, because he really helped me a lot to improve my theoretical background. We had like discussions for hours and hours, and I would used to ask naive questions because I was new in the field, and he would patiently answer and explain everything to me. And yeah, so I'm really, I think I was lucky.
It's good, but it also means you could get a nice opportunity, and it's a new opportunity also for someone else now, the next one who will join the lab. And I'm sure you will also have plenty of opportunities in your postdoc and in your future career, which is great. I think there's no more question, and we're almost at the end of the session. So except if there's one last minute question, or you feel you really want to ask this question. Otherwise, I think you can contact Tariq also if you have some question you didn't want to ask live.
Yeah, sure.
Otherwise, thank you everyone for participating. Thank you very much, Tariq. It was a very nice, clear presentation, a very nice work, impressive work. So well done. And good luck in the future. And we hope to hear from you on your future project, and also maybe on this project from Jean-Christophe Jonas' lab, about all the next part of the data which will be acquired on this project. Thank you very much. And I hope we will see you soon on the next activities from the EASD Academy. So we will have a webinar in May. So, and then another journal club in a couple of months. So subscribe to our newsletter if you haven't done it yet, and you will get all the information. Otherwise, everything is on our website, on Twitter, on Instagram. So keep updated, and hope to see you soon. Thank you very much.